sclerotia that are point sources of contamination. And I mentioned already, so these chemical compounds, these ergot alkaloids, they can cause health effects. Some of them may be good. In fact, some of these compounds or derivatives of these compounds are used to have pharmacological uses in medicine. Migraine treatment is one of them. But in general, we're looking at these from a safety point of view and mitigating risk to consumers. So one impact of these compounds is their vasoconstrictors, so they reduce blood flow. So if you can think of in terms of, say, if these compounds make their way into animal feed, and this happened early 2000s, mid 2000s, again in Western Canada, there was some contaminated cattle feed. So the impacts then are gangrene and loss of limbs for the livestock. And then just to reiterate too, this is not, the safety aspect is not just for food, for humans, for people, for our food products, but it's also for feed. Because the grain handlers, we're within the grain handling chain. The ergot sclerotia can be cleaned out. They're a different density, they're a different color. But sometimes what happens is the grain screenings, or even if grain is downgraded and the downgraded grain may be diverted to feed channels away from food, we still have to be, I guess, cognizant of the health risks. I mentioned downgrading briefly. So ergot is actually a grading factor in Canada. So what that means is when grain is delivered to a terminal, or when grain is delivered to a grain elevator or loaded onto a vessel for export, inspectors assess the grain for the presence of ergot sclerotia. And this is done visually. From speaking with the inspectors, it's a fairly easy or simple grading factor compared to some of the other ones that they're looking at, because the sclerotia are dark, very different from healthy kernels. So visually, they're easily discerned from healthy kernels. So in Canada, depending on the grain, there are tolerances set for the amount of ergot sclerotia that can be present in the grain. And if the tolerances are exceeded, then grain is downgraded. And that potentially, if it's a producer delivering, that can mean a loss of money for the producer. If it's within the grain handling chain, then that means that maybe the grain handlers have to spend extra time and resources to clean the grain or somehow deal with the presence of these sclerotia. So the purple table is showing the Canadian tolerances just as an example for Durham. You can see for the higher grades, it's only a limit of 0.02% by mass. And then for lower grades, lower quality, the tolerance is a bit higher. And then the blue table here, there's also some tolerances on kind of the international scene for ergot sclerotia. Codex Alimentarius sets regulations to govern international trade, and their limit of ergot sclerotia in wheat, not just Durham, but all wheat, is 0.05%. Same thing with the US. And then I think a bit more timeliness, a bit maybe to emphasize the relevance of this symposium, the EU at the end of August posted an update reducing their tolerance for ergot sclerotia to 0.02% by mass in grain. And that's coming into effect January 1st, 2022. So I mentioned briefly kind of the introduction for the Canadian Grain Commission and earlier on how we do a number of monitoring and surveillance activities. Ergot itself is monitored by our Canadian Grain Commission inspectors in samples that represent the large exports, the cargoes, as grain is loaded onto vessels. In the GRL, so my group, we're monitoring randomly selected shipments for ergot alkaloids as well. So we have the inspectors looking for the sclerotia, and then we're also doing complementary work on looking at the presence of the ergot alkaloids in the grain. And the graph here at the bottom is a summary of some work that we did a few years ago. The orange boxes all to the left with the grades and the feed grade, the numerical grades and feed grade, those all represent, those all summarize export cargoes. So this is, if you can picture this, this is at the end of the grain handling chain. Grain has been delivered by producers, it's been handled and managed for quality factors as well as for safety factors by the grain handlers, and then loaded onto a vessel for shipment elsewhere. And then the green box is a couple of years of harvest survey data. And these would represent samples at the beginning of the grain handling chain. So these are samples that are taken by producers from their harvest. So right at the very beginning of things. So you can see that there's a wide range of ergot alkaloids present in the harvest samples. But when we look at all of those other shipments, where we look at the shipments to the left, all the orange boxes, the concentrations of ergot alkaloids are quite small. So for me, the take-home message here is that the grain handling system is managing the presence of ergot sclerotia and ergot alkaloids in the grain, but with challenges as things are changing from year to year. And if the presence of ergot and infection by Claviceps becomes more prevalent or even more serious, then there could be challenges for the grain handlers in ensuring that they can keep ergot and ergot alkaloid concentrations low. And I think I'm going to finish off here, Sean, with one, this is kind of a near and dear to my heart research, but sampling is key for assessing ergot. Whether you're looking at sclerotia or you're looking at ergot alkaloid, sampling is key for anything, but especially for this kind of stuff, where if you think back to what I said at the beginning, how the ergot alkaloids and the ergot themselves, we can consider them to be point sources of contamination. So they're less dense than healthy kernels. So as grain is moved and handled, they can separate out. And we have a very heterogeneous distribution of these contaminants in bulk grain. So we need very good sampling equipment and procedures to ensure we have accurate and precise measurements, both for the assessment of ergot sclerotia and for the assessment of ergot alkaloid. So I'm not going to really speak more about that, but we have done a lot of research and continuing to do work on this and developing good practices so that the numbers we end up with at the end of what we do, there's something that we can put value on and rely on. So thank you. And I think I'm going to hand it off here to Sean now.
Yeah, thanks, Cheryl. And Cheryl brings up a good point about sampling. And as Jim mentioned, we have a program called the Harvest Survey Program, and that allows us to do a lot of sampling of grain across the country for different grading factors, including ergot. So the Harvest Survey Program is a voluntary program. It's been going on for many years. We have data going back to 1995, where producers submit grain samples to the Grain Commission, and then the scientific staff at the GRL, as well as our inspectors, will go through those samples and measure different factors that would be important to the producers. And then we report back to the producers on some of those so that they have an idea of what their harvest looks like. So on the bottom left here, this is an example of an envelope that would be received by a producer, and they fill that with a sample of their grain, and we give them instructions on how to properly sample that so that they can send that to us. It just goes through regular mail, and then it goes to our headquarters downtown in Winnipeg, where the analytical services team within the GRL processes them, and then it goes to inspectors who can go through the grain and pick out things like individual infected ergot sclerotia. So this is a bit of a snapshot of the ergot data that we've been collecting throughout the years. So this is on the top here, some trends might pop out to you a little bit immediately. You see this blue line that seems to be higher than the rest, and this corresponds here to rye. So rye is particularly susceptible to ergot infection, and you can see that the percent of samples of rye that have measurable amounts of ergot is much higher than that of the other cereals that we investigated in the survey. And this is sort of followed by bread and durum wheat. And then for barley and oat, we see that these tend to remain quite low. Another trend that you might see just visually by looking at this is there's a lot of up and down, sort of like what Jim said. You know, it's not the same every year. It does go up and down. You can see sometimes that can be quite drastic in some years. But there does appear to be some trends, and Jim was mentioning maybe in the late 1990s, he started to see some increased interest in this because there's more ergot popping up. Looking at our data, we see maybe a clear division sort of from 2010 onward. So what I broke up our data set from 1995 to 2010, and then 2010 onward. And you can see the table here with some of the summary statistics. So this just sort of gives you an idea of the scope of the harvest survey program here in terms of numbers. So we're receiving, over 10,000 samples per year across different grains. So this is it's a massive, massive survey. So we have, over 100,000 bread wheat samples just from 1995 to 2010. 4.2% of the samples had a measurable amount of ergot in them during that time period. And then if you look at the same bread wheat samples, but from 2010 to 2020, you see this increase to 19.5% of samples having a measurable level of ergot. So this sort of supports what Jim was mentioning, where we're seeing more incidence or occurrence of these. But what's interesting is if you look at the median amount of ergot in those infected samples, it hasn't changed a whole lot. So we're seeing it sitting around 0.02% in both time periods. So although we're seeing maybe increased occurrence of ergot perhaps across the years, if they are to have ergot, the amount of ergot in those samples doesn't seem to be changing too much, which is interesting. If we move down to the next row here to Durham wheat, again, large sample sizes here, and we're going from around 3% to around 13% with no change in the amount of ergot if they are infected. For barley, prior to 2010, there wasn't a whole lot going on. We have 0.3%, and I don't report the median percent of ergot because 0.3% is one sample out of that 394. So low occurrence of in barley prior to 2010, but then we do have some samples popping up after that, but the levels overall remain quite low. Contrast to rye, where we're seeing 27 and 65% of the samples being containing some measurable level of ergot. But again, the levels aren't really changing within those samples. So if they have ergot in there, the amount of ergot isn't changing a whole lot. And then we only have a couple of years of oat data, most recently in the last couple of years. And overall, we didn't see a whole lot of ergot in those. And if they did have ergot, there wasn't a whole lot in there. So to go back to Cheryl's point, though, and that's that the grain handling system seems to be able to cope with this quite well. But we do see sort of a correlation between the amount of ergot that we're seeing at the harvest level and the amount that we're seeing in the cargoes. So rye, for example, seems to have higher levels of ergot alkaloids in the cargo samples, followed by bread and durum wheat, and barley and oat remain quite low. So this is just to say that the grain handling system seems to manage ergot quite well, but there is some ergot that appears in cargoes, and these seem to reflect the levels that we see in harvest. So we can actually dig a little bit deeper into each of the crops and what's going on across different provinces. So shown here are the trends across provinces for durum wheat. We can see that we don't really see any durum growing in Manitoba past 2010. There used to be more durum being grown in that province. But generally, we see that sort of break at 2010. And then maybe some differences between the provinces, but it's highly variable. Perhaps Alberta is a bit higher, but it's kind of hard to tell. If we're looking at the level of the number of infected sclerotia within ergot samples that have ergot, we see that most of the samples fall within this blue and this orange range, which are actually below the levels for even grade level 1. So these would be considered high grade, even though they have some level of ergot, whereas grain below would be ones that might affect grade as well as quality. And you can see that proportionally, these are a few of the samples, not the majority of those that actually contain ergot. So ergot levels are still, quite low even at the harvest level before they're cleaned. If we were to switch over and look at bread wheat, we see some similar trends, lots of variability across years, across provinces. There's some variability, but again, maybe Alberta is a little bit higher than the rest, but in some years, other provinces seem to be higher. So it's perhaps hard to tell. And again, similar sort of results in terms of the percent of ergot within those samples, where the majority are sort of within these tolerable levels that would not impact grade, where there's a small subset of the samples that would be impacted. With bread wheat, we have lots of samples. Like I said, over 170,000 samples. So we can actually look even closer than the province level and start to dig into individual regions within provinces. So shown here are the three main prairie provinces where we grow bread wheat, and we can subdivide these into different crop districts. And we can look at the percent incidence or occurrence of ergot within each one of those crop districts. So again, you're seeing the same trend across years, perhaps maybe 2010, where there's more red over here, suggesting increased incidence or occurrence. There seems to be some variability within crop regions, crop districts, but no crop district seems to be immune. This seems to be sort of widely distributed across the prairies, but maybe there's some hot spots in some provinces. You can see here Alberta in 2020 was quite bad. And then in some crop districts, for example, Saskatchewan 1A, which is sort of this southeastern part of Saskatchewan, is maybe hit a little bit harder than the rest in that particular year. So there is some regional variability. And then Cheryl's group did some follow-up of ergot alkaloids in many of these different regions, and these seem to parallel what we were observing in the harvest survey. So I sort of highlighted two red boxes here that were particularly high for the amount of ergot alkaloids that were present. You can see one of them is this region 4, which is southeastern Saskatchewan, where we saw that sort of redder crop district in the previous slide. And again, Alberta seems to perhaps have a little bit more, and these seem to be higher in levels that have a reduced grade, which may have been in part due to ergot, but could be caused by other factors as well. Whereas, the number 1 grade had reduced levels of ergot alkaloids. So to summarize what Cheryl and I have presented to you today, so ergot is a fungal disease. It's been infecting cereals for centuries, but maybe there could be increased occurrence, particularly in the last decade or so. Ergot occurrence affects grain quality and end-use properties. We saw that in the semolina as well as in the pasta. Then the toxins also have a grain safety component to them. So it's important to monitor for those. And the level of ergot and ergot toxins seem to vary by crop, by grade, by year, by province, by region and district. So this sort of highlights the importance of monitoring for ergot, both in cargoes and in harvest, and how we're actually quite fortunate at the CGC that we've been maintaining good historical records of these, and plan to continue doing that going forward. Because these are important for guiding both crop and management practices, policy making, and how the grain handling system should be coping with these. I'd like to end by acknowledging the hard work done by the CGC Grain Research Laboratory, Cheryl's team, the Trace Organics and Trace Elements team, my team, the Microbiology and Grain Genomics team, but as well as the other teams within our group. Cheryl showed some data from Bin Chao Fu, who does the wheat quality aspects, but our analytical services team, which manages the harvest survey program, is particularly important. Carrie, who's the current manager of that program, and also did a lot of toxin work with Cheryl, and Twyla, who managed that program for several years. And this is all a highly cohesive effort with other departments and divisions within the department. So the industry services that looks at those infected sclerotia within the samples and provides that data. And last but certainly not least, Canadian producers. I think they work harder than all of us. They're feeding the country and the world. And yeah, I'll end it there, and I'll Be happy to answer questions, or I think I'm not sure if Jim wants to transition directly into the next presentation. I'll turn it back to you, Jim.
Okay, if you could stop sharing your screen there, Sean, that would be great. I think what I will do is I think I will move on to the next presentation. But thank you, Cheryl and Sean, for the presentation. I mean, the work that's done by the Canadian Grain Commission is pretty important, and it provides us with a lot of data on what is actually going on in the farmers' fields. And it's a resource that is often overlooked. So I think in Canada, we're actually very lucky to have that kind of work going on. And I guess pass on our thanks to the Canadian Grain Commission, Sean and Cheryl, for the work that you've done. So we'll move on to the next presentation, and we will have questions for Cheryl and Sean after this next presentation, and at the end as well. So the next presentation is entitled Transcriptome Analysis of the Wheat Claviceps Interaction from Both Perspectives. And this is being presented by Dr. Anna Gordon of the National Institute of Agricultural Botany in Cambridge in the UK. She's worked there for the last 15 years in wheat fungal interactions. And unfortunately for her, a good part of that time she has been working with me, and it's been a real pleasure to work with her. She has been a great collaborator. So her work there in this regard is looking at the wheat-ergot interactions and particular resistance to ergot, and more recently in the transcriptome analysis area go. A year ago, she became the facilities lab manager for the National Institute of Agricultural Botany, but indicates she still maintains a love for Claviceps. So Anna, if you would like to share your screen, and away we go.
All right, bear with me. Right. Hi there, and thanks, Jim, for that lovely introduction. I must say you've been a brilliant collaborator too. I'm assuming that everyone can hear me and that it's all, and you can see my screen, so fingers crossed. So, yes. Let me go to the next. How do I know go to the next slide? Does anyone want to come off mute and tell me what on earth I'm doing wrong? I'm doing my normal.
Are you using multiple screen or?
Pardon? Can you see my tool?
Screen, yeah. Yeah.
Okay, I'm having a problem moving it on from the. Oh, we've done it. Sorry, I had to do a left-hand click rather than right. Fingers crossed this will work now. Yeah, so I'm based at NIAB in Cambridge, and it's changed a bit since Jim was last here, in that our building has now massively expanded and we've got a new upgraded facility. We've also got a partnership with the University of Cambridge with the creation of the Crop Science Centre, which is the building towards the back. So we're hoping that this is going to lead to quite fruitful collaborations in the future. The other picture I've got there is our Park Farm site, which is a couple of miles away, just to show that we've got a large amount of glass and also lots of space for field trials and all the rest of it. So what I thought I would talk to you today about is to give you a bit of an introduction to the UK perspective, and then to touch on 3 different aspects of our most recent sort of work, maybe talk a bit about the future. So it's been a bit of a strange time in the UK and the rest of the world, what with COVID and stuff, and it seems to have taken quite a long time to get some of this stuff published, but 2 papers that are still in press, we still don't quite have them out there, I'm going to be talking about some of the key findings that we found in those today. So in a similar way to what you find in Canada and probably the rest of the world, the main reason for Jane rejection at UK maltsters and millers is ergot and the presence of ergot. So despite advances in separation technology, there's an increasing acknowledgement that grain can be contaminated even in the absence of visible sclerotia. And this has led to quite a major change in the EU states. So Cheryl mentioned that there's a new EU regulation to do with quantities of ergots that are allowable in grain. So it came out at the end of August, and not only this, for the first time, they will be monitoring ergot, the presence and quantity of ergot alkaloids with quite strict new limits. So, so this is the quantities that are going to be allowable. So for unprocessed cereals, again, so this is the sort of equivalent to what you've got already. So it's a 0.2% in wheat, barley, etc. But for rye, these levels are higher. But if you notice here that actually quite a major change that's going to come in is that after 2024, and probably depending on how the next couple of years go, but actually they're going to be much more stringent limits to do with the amount of sclerotia that are allowed. And the other thing to say is that the ergot alkaloids themselves, so this will be the sum of the alkaloids, and these values are similar to what Cheryl and Sean was showing. These are micrograms per kilograms or parts per billion. So when I talk about this in my talk, I'll be using probably parts per billion, but this is what it's equivalent to. Again, a lot of these levels are going to increase after 2024, so it's going to have quite a big impact, I think, in UK, in agriculture in the EU. And for us in the UK, it will be in ways we don't even know yet, because of Boris Johnson's omnishambles of Brexit, COVID, disaster. We still don't really know what's going to happen with EU regulations and how they're going to affect UK farmers. So we're still sort of waiting to find out, really. So anyway, onto some science. So this first one I want to talk about, it's basically, it was a big transcriptome experiment that we did, basically looking at transcriptomic changes that happen in wheat in the first 7 days of an infection with ergot. And so these pictures I've put on just to show you, basically we excised ovules like this. We then dissected them, and then at various stages after inoculation, we then harvested that RNA. And I just was going to take you through the stages that we took. So one was 24 hours, where all that you basically get are hyphae within the stigma. And then we've got 48 hours where you've got the first hyphae coming in. We took 3 days, which is where you're basically beginning to see hyphae down the bottom of the ovule. And then this picture is actually from 5 days, where you're getting almost total collapse of the tissue because the fungal structures have basically overtaken any of the wheat. And we also took a 7-day, but they were completely demolished by the fungus by that point. So what we did, we collected RNA, we Illumina sequenced it, and then we mapped onto the wheat reference sequence genome, and we did a differential expression analysis. And so the next couple of slides I show are basically going to be the sort of summary of basically what we found. So I'm going to briefly touch on a defense response, alkaloids and hormones. So this graph is basically looking at log2 fold changes in in ovule tissue from 24 hours up until 7 days. And what we've done is we've split it into 3 groups. So this was the stigma, of which it basically was only intact enough at 24 hours. So we've only got 1 time point for the stigma, but for the transmitting tissue and base, which are, we basically cut the ovule in half. So for these 2 tissues, we've then got basically from 24 hours up to 7 days. And so the big take-home message here is, yes, the defense response is triggered. For a number of years, we were sort of toying with this idea that possibly, you know, Claviceps imitates a pollen grain, you know, it goes down the same tissues, it doesn't trigger a defense response, there is no known resistance. And I'd say that this pretty much refutes it, in that all of these different categories of defense response are pretty much upregulated in response to the presence of the fungus. And interestingly, so with stigma tissues that were taken at 24 hours after inoculation, as you can see, quite a few of these have got genes that are upregulated at 24 hours. So it's actually a really quite early response. And I'm going to just touch on something really interesting, because I think there are some transcriptional changes that are triggered away from where the fungus is. So what I'm going to, so this 24-hour time period. So if you look at the, oh gosh, keep losing these, NBS-LLR proteins, they're on at 24 hours in the stigma tissue, as are the receptor protein kinases. But then really interestingly is that at that same time point, at 24 hours after inoculation, you can see that there is a difference. Okay, so compared to the mock, the inoculated are either downregulated or upregulated. So what we're suggesting is that there must be some kind of signal that goes from the stigma, which is interacting with the fungal tissues, into the heart of the ovule, where the fungus has not yet got to. And all of these are really interesting, suggesting that there's a mobile signal. Secondly, I just wanted to touch really briefly on the Ergot alkaloid genes, because as many of you probably know, the Ergot alkaloid genes are contained in a cluster in the Claviceps genome. And so as well as mapping to wheat, we've also mapped to the published Claviceps genome. And one of the things that we noticed was that even in the stigma at 24 hours, there were actually quite a few of these genes that were already present. And by the base tissue at 7 days, so by this point, it's well and truly colonized by Claviceps, all of the alkaloid genes have been turned on. So this might explain why at such an early stage, so in honeydew, why you get base levels of alkaloids, even though they're there at a small amount. Thirdly, I'm just going to touch really quickly on the hormones. So all of these genes are either ones that are coding for the biosynthetic pathway, the signaling pathway, or the degradation pathway. And so as you can see, quite a few of these classes have actually go up as the infection progresses, and then they plummet downwards. The reason why all of these plummet downwards, I should probably have said before, is basically because the wheat tissue is replaced by fungal tissue. So when you're comparing the inoculated to the mock, that's why it looks like everything plummets by 7 days. It's true, because it's been taken over by the fungus. But it still gives us a bit of an early indication of the fact that there are various hormone pathways which are being triggered. And I just wanted to pull your attention again to the fact that at 24 hours, the jasmonic acid, there are some genes that are already present and triggered in the stigma tissue compared to the mock. And I wanted to just say that not only the jasmonic acid, but the ethylene too, are also being upregulated in the base tissue. So this is, again, is a couple of millimeters away from where those fungal tissues have got to yet. And I think it's really interesting. When we looked further at what the ethylene genes were, it looked like quite a few genes have got ethylene responsive factors. So what we're proposing, therefore, is this hypothesis that it could well be ethylene is that mobile signal that is then triggering a defense and hormonal response in the base tissue. So I'm sorry to sort of skip around quite so quickly, but I'm actually going to carry on with this hormone idea. So a number of years ago, we published our findings where we identified QTL that had an effect on ergot sclerosicides in a UK mapping population, where we found that ergot sclerosicides co-located with mutant DELA genes. These are the reduced height loci. And we found that the double dwarf lines within that population, where they had 2 copies of the gibberellic acid insensitive DELAs, they produced smaller sclerotia. And so we sort of wanted to follow it up by looking at more RHT mutants to see if it corroborated this. So there are a number of lines that we managed to get hold of thanks to some collaborators at the John Innes Centre. So one is in the Mercia background and one is in one called Maris Huntsman. Now these are really, really tall, great things, but there are a number of mutant lines that we could get hold of that also have mutations at other places within those same DELA proteins. And we also included, for reference, RHT8, which is a gibberellic acid sensitive mutant, and also RHT12, which has nothing to do with gibberellic acid, it's steroid pathway. So we did a glasshouse-based study. We had these RHT mutants and we wanted to compare them to the wild type. So some plants were allowed to produce more mature ergot, which is what led to these graphs here of the kind of relative weights. And other ears that were inoculated at the same time, we sampled for hormone analysis taken at 3 days after inoculation. And so just on these graphs here, what we found was that the wild type have got quite a wide, broad range of weights, as have some of the single semi-dwarfs. So if you have one of the dwarfing alleles on its own, it doesn't really have much effect on the weight of the sclerotia. But the severe dwarfs, which are equivalent to the double dwarfs, as in B1C and D1C, had a much smaller sclerotia. So it backed up what we'd found in our study. So just the first bit that I want to show you is just to show you that when we sampled for active gibberellins, well, we just sampled for one, we sampled for GA4. And these graphs are basically showing the control compared to the inoculated. So the control are uninoculated. And I think what's the first thing to say is that upon infection with Claviceps, you get big increases in active gibberellins in that of your tissue. What's interesting as well, I think, is that in the background of Mercia, all of the lines with the Della mutations have got quite a large, a raised gibberellic acid content in the non-inoculated as well. This is probably because these lines are partially, if not quite a lot, insensitive to gibberellic acid. So I think when you're sort of comparing the weight and the gibberellic acid, what we're proposing is that it's disrupted the natural progression of Claviceps. It's for some reason, it's not as successful as it would have been, which results in smaller sclerotia. And finally, we also looked at auxins, which is even more apparent that these tissues are absolutely flooded with auxin upon infection with Claviceps. And these actually corroborate what we found with the transcriptomics results as well, in that the most upregulated gene family out of everything in response to Claviceps infection is an amino acid synthetase. And these code for proteins which sequester or mop up active IAAs from cells. And interestingly, again, it's the extreme dwarfs. Sorry, my cursor's moved. So these extreme dwarfs, the B1C and the D1C, are the ones that produce the really smallest sclerotia. Actually, they've got a much smaller relative increase of IAA. And I would say it points towards further unknown mechanisms of the interplay between these two different hormonal pathways and the responses. It's probably worth saying we don't know if these hormones are being produced by the fungus or the plant. We know that there are likely to be IAA biosynthesis genes in the fungus. And so what we're hoping is that when we elucidate more about what those genes actually are, we'll be able to back it up with the transcriptomics to say that it's likely that Claviceps is producing, it could well be producing auxins, which I think has been kind of known about, but as far as I know, it's not been published yet. So, and that brings me, hopefully, I've got a couple more minutes, I'm going to whiz through it. So I just wanted to touch on a bit of stuff to do with alkaloids and are they moving within the plant. So this was part of an AHDB funded study that we did a couple of years ago. It's not made it, I mean, it's available on the AHDB website, but we've not actually published it in full yet. But I wanted to point to this, one of the experiments that I did there, where we basically wanted to test to see if ergot alkaloids can move within the developing ear. And so to do this, we had an inoculation site where we just literally did either 4 or 8 ovules. And then we very carefully left it all to mature. And then we separated the grain that formed at the top and the grain that formed at the bottom, and then got those tested for ergot alkaloids. And apologies that this is quite a messy graph, but what I basically wanted to show that each of these is an individual rep. So barley, grain site above the infection and below the infection, it had whacking great amounts of combined ergot alkaloids. And you can see you get quite a lot of variability within reps, which means it is quite messy data, but I would say it's a really interesting first look at something that might be happening out there in the fields. When we had a look at those ergot alkaloids, if you compare the ergot alkaloids that we found in the sclerotia from those same ears it was literally just 4 ergot alkaloids that were detected. Whereas if you look at what happened in the combined ergot alkaloids in grain, so those that were formed above a sclerotia for wheat, barley and rye, and those that were formed below, I think it's quite apparent actually there's a much more diverse range of alkaloids that appear to be within the grain itself. Quite interesting. So what I'm kind of suggesting here, so this is kind of a schematic of a cross section of an ovule in a rachis. And so what you have to imagine here is that this one's been infected. And so what we found that at 7 days, you know, it's all become, it's turning into, the name's gone, an immature ergot. It's become a very porous kind of structure. It's beginning to ooze honeydew. And basically that is being driven by the pressure of water and phloem sap that is basically coming from the vascular tissue. And so not only is this feeding the fungus and meaning that it can produce these conidia, which then go on and are exuded from the glooms as honeydew, but what we're proposing is that some of these alkaloids are actually making their way back into the phloem. And I say the phloem because a lot of these alkaloids are non-water soluble, which means that I think that they would require active transport in order to move around. So this is an interesting first step, and I think it's an experiment that I'd very much like to see repeated, possibly as a PhD proposal. So what role could alkaloids be playing? Is it protection against herbivory, or do we think that they have a role in pathogenicity? That's kind of, we don't know yet. So that, oh, just really quickly, I know I've run out of time. My final slide was just to say that as part of that same study, we took a number of actual ergot sclerotia from 7 different ergot strains. Four of them were from the UK. This one was from Germany, and these 2 were from Canada, thanks to Jim and co. And I think it's kind of quite apparent, really. They have quite a diverse makeup of EAs. And what would be interesting to know is this stable year after year? Can we begin to unpick what the genetics are in these lines that might be influencing this relative quantities? And I'm hoping that through future collaborations and sharing of data sets that we might get to grips with some of this side as well. So that just lets me say, I'd like to say thank you to Nelzo, who did all of the bioinformatics. Eleni was my PhD student, has been an absolute star. She started writing her thesis at the beginning of lockdown and had done it in 6 months, and hats off to her because it was a really difficult time. I'll say Donal O'Sullivan because he was the originator of a lot of this work, and Leslie Boyd, who still works at NIAB, is hoping to carry some of this work forward with the breeders and hopefully collaborators. So thank you very much.
Thank you very much, Anna. So I'm going to open it up to some questions from anybody. They can either write it in the chat, or you can unmute your microphone and ask the question. And this will be for Cheryl, Sean, or Anna, whoever has questions. And I can start it off for all 3 of you. So with your work with the ergot alkaloids, I'm curious about how are the standards for ergot alkaloids in grain evolving And that could be in the EU and Canada. So I'll start it off with Cheryl, since she's the mycotoxin expert. I'm picking on you, Cheryl.
No problem. So how are the standards evolving? So I'm assuming you're talking about the regulations, the tolerances. Yeah. So I mentioned, and then Anna mentioned afterwards, that really the latest activity has been in the EU, where they've lowered their limit for ergot sclerotia in grain and introduced for the first time a limit on ergot alkaloids in grain. So no, I'm not aware of any other jurisdiction. I think way back Uruguay might have had a posted limit for ergot alkaloids, but nothing from international bodies like Codex. So just from experience in other contaminants, food contaminants and grain contaminants, I think we're moving towards stricter limits or maybe more widespread limits. I'm not aware of anything of Health Canada moving forward with any assessments or exploring the potential need for regulatory action on setting Canadian limits. I could see down the road, perhaps the next step would be Codex discussing this at an international level.
Okay, Sean or Anna, do you want to comment on that, or are you good with Cheryl's explanation? I'm sure it's pretty good.
Yeah, it is. Yeah, it is. What I don't really get to grips with is how they're actually going to be monitoring it, because the millers and masters are basically in a panic, because they say, you know, it's 3 days between grain going in and it appearing in bread on the shelves, and how on earth are we going to stop production in order for them to test it? Or is it all going to be tested at bread at the other end? So there are so many uncertainties about how this EU regulation is actually going to play out practically. It's got people really worried.
Okay, so we have a question here for Anna, actually two. What are the roles of ergot alkaloids in the infection process inside the host plant? And why do plants keep feeding the infected part?
So, do you think that Claviceps is just a symbiotic partner with wheat, aiming to limit herbivory or us eating it? It could be that it's got so many evolutionary links to other symbionts of similar class in the Clavicipitaceae. That could be one thing. We don't have any evidence that it has any pathogenicity to the plant. So we just don't know what the point of it is. As far as we know, it's not got a point in the infection process, but we just don't know. Okay. Why do plants keep feeding it? I mean, I think the plant loses the ability to sever the vasculature feeding it, and so it just keeps on pumping it out, because it's like an open tap, whereas the rest of the grain sites within that ear are all blocked because they have a developing grain. Actually, the ergot one, it's like a sponge. It basically just flows through it. So whether that's just a physiological thing, I don't know. There you go.
Yeah, I will admit, in part of your talk, you were talking about the alkaloid production and wanting to know with the different isolates, is it stable year after year? I find that an interesting question, because I also think just looking at the genome or the genetic structure of each of these isolates, and it, you know, to me, it'd be interesting to actually infect plants and over a period of time induce them to form ascospores and then reinfect and do that over a number of generations and see whether the, you know, how much of the genetics of each isolate changes if it does. I think there are some interesting questions along that line, and I suspect it would all be related. What do you think, Anna?
I reckon so. I'm hoping that Mindy later on is going to elucidate what differences are between some of these strains. It might be a bit early days to ask for that sort of thing, but it's definitely going to help us find out.
I would just stay tuned until 11:30. We're reporting on some pan-genome work that a grad student had done, and you'll see that there is lots of what we call lineage-specific genes that aren't core to all isolates, but there's lots of variations. Things are present and absent in different strains, and that may be leading to this variation in the toxins that you reported between strains.
Thanks, Kurt. I do look forward to that. And just in case people are wondering, I'm kind of focusing on questions for Anna here because I'm hoping Cheryl and Sean will stick around to the end, in which case I mean, they can be asked questions as well. So another one here is very nice work, and you've shown that infected tissues have higher levels of auxin compared to the mock. So does the pathogen produce any auxin, or is this totally induced in the host? And is there any antagonism between auxin and cytokinin?
Oh, I'm in the dark about the auxin and cytokinin. We did look for levels of cytokinin. And off the top of my head, I don't think that they were significantly different between the inoculated and non-inoculated. But whether the fungus is producing auxin, I think it could well be. I mean, I must say it was the sort of like Friday afternoon job where you look at related fungus, what the genes are they present in Claviceps? Yes. Are they upregulated in these tissues? Yes. But what you don't have, I guess, is a definitive labeled, you know, you could do some really nice experiments to prove yes or no, which would be really nice to do.
Okay, here's one for Cheryl and Sean. So this one is actually a question that I've asked them a number of times. So what's changed in recent years that's caused ergot to increase? Is it involved with is it related to the environment, the host, or the pathogen? And similar along that lines, do you also see the proportions of the alkaloids change with different crop years? So I'll let Cheryl and Sean fight over who gets to answer these.
Maybe I'll tackle the first bit if you could tackle the second bit there, Cheryl.
Yep.
So I think that, you know, some of this we've discussed a little bit already, that, you know, there are differences in genetics. So I think what we're looking at is a combination of multiple factors, right? And it might be hard to pin it down on a single one. We have these genotype both on the host and the pathogen side, by management, by environment interactions, and these are sort of always changing. And depending on the year and the situation, you're going to have a different level of ergot. And, you know, in a given year, it might be hard to pin it down on a single one. But certainly over the last decade or a couple decades, we've definitely seen differences in management practices in Canada. In terms of genetics and on the pathogen side, I think more work needs to be done. So I'm excited for the talk this afternoon to see what they've uncovered there. On the host side, I don't think that there's been perhaps enough emphasis on breeding for ergot resistance. It is sort of on the radar, but in Canada, it's probably not even second or third fiddle to, you know, yield and fusarium and some other targeted traits for crop improvement. And environment, you know, we're looking at climate change, we're looking at all sorts of other things that could be impacting this as well. So definitely lots of work to be done. So it'll keep us gainfully employed for years to come, probably. And I'll move to Cheryl for maybe the toxin aspect of that question.
Sure, thanks. I think just maybe just to add a couple other examples, Sean, you mentioned management practices. I think what's changed as well, and I'm sure there's other people on the call that are better informed about this than I am, but things like moving towards no-till or having increased buffer zones around fields where you have kind of a, I don't know if a sink is the right word, but you have these, places where the Claviceps can reside. With regards to changing patterns over the years, I'm thinking to some of our work we've done on harvest samples and for cargoes. We don't really see the changes in the patterns from year to year. But when I consider the samples and the data that we're looking at, we're really looking at kind of the end of the line with respect to the cargoes. So we've had a lot of mixing happen. There's grain coming from different sources. So it could be that there are minor changes, but they're just kind of smeared out when we mix everything together. Where we really see the changes, and I think Anna showed a slide. We have similar slides from work that we did with Jim, where we see the changes amongst host plants and isolates. So I guess when we really break apart and focus on those factors that can affect the pattern, that's where we see the difference. But when you're talking about bulk grain and everything's all mixed together, we don't see differences. The other point where we do see differences in almost, you can consider it almost like the freshness of the grain. When we look at harvest samples versus cargo samples. So the cargoes, by the time we analyze them, or the time that they're moved from, say, on-farm storage to grain elevators to terminal elevators and loaded on a vessel, and the sample makes its way to us, could be years down the road, depending upon what the farmer's doing, what price is doing. We see changes with, I think now we're talking about physical chemical properties and changes to the actual chemical structure, just from whether that's exposure to light or just time. And chemically, something's happening where we're seeing interconversion of some of these compounds. So hopefully that answers the question there.
It would be interesting to know, Cheryl, if any of our samples overlapped with yours, to see if it gave the similar alkaloid profile. Might be.
Yeah, we can look at that. I know I saw in your graph you had EI2 and EI4. I know that's, Jim would probably be able to kind of indicate which two we should be comparing, but absolutely we should do that.
Okay, well, what I'm going to do right now is I'm going to thank Anna for showing up. I know she has other responsibilities she has to do this evening in the UK. So thank you, Anna. And what I'm going to do is give everybody a 15-minute break. So we'll come back in 15 minutes. And I'm sorry I interrupted you.
Thank you. Really nice to see you all.
Okay, so take care, everyone, and we'll see you in 15 minutes.
Bye.
So good morning, everyone. I guess we're getting back to our time to begin the second part of this workshop. Jenna, are you having trouble? Are you able to see or hear me? Yeah. I'm hoping that you'll be able to see the presentation as it comes up. So the next presentation that we're going to be doing is being presented by Meow or Mindy Liu. It's entitled The 168-Year Taxonomy of Claviceps in the Light of Variations from 3 morphological species to 4 sections based on multi-gene phylogenies. So Meow is a research scientist with Agriculture and Agri-Food Canada at the Ottawa Research and Development Centre. She works in the Biodiversity and Bioresources group with Agriculture Canada. And her research interests include taxonomy, phylogenetics, molecular diagnostics, population evolution, genomics of plant-related fungi. Currently, her research is focusing on the powdery mildews, the ergot fungi, and grass endophytes such as epicloe. It's important when we start looking at these things that, as a plant pathologist, that we realize that we really do have to know what we're working with. And sometimes as taxonomy changes, we have to make sure we keep up with these things. So with that, I will turn it over to Meow. And if you want to share your screen, Meow, you can begin your presentation.
So did you see my screen? So you see my screen?
Yes, so we see the acknowledgements. Yep.
Okay, yes. So thanks, Jim, and for organizing this, give us an opportunity to share our research. And so first of all, I really want to take this opportunity to thank all the people who have helped me during these past 5 years. Especially when I just started this project, Jim and Keith handed me a big set of cultures and sclerotia I can start with. It's really fortunate. So we can start with something to work with. And then after that, friends, colleagues, and institutes, collaborators, and they all provided research material, and that's very fortunate. And I also get good technical support from our wonderful technicians and MTL at ORDC, and also a whole bunch of students all contributed to this research. And we mainly focus on taxonomy, but meanwhile, we collaborate with other people, so like Kirk, he will give a presentation later, and also Anna, and gave presentation just now, and on other aspects of the study. And we also have a collaborator with bioinformatic expertise and chemistry expertise working on the genomics and metabolomics, and our research was funded by our ABASE. And this is just very fortunate, and have human resource, have financial resource, everything is ready for me, and I'm very fortunate. So today, sorry, I will just give a very brief overview of the 168-year taxonomy of Claviceps in the light of variations from 3 morphological species to 4 sections based on multi-gene phylogenies. So I separated the history to two time periods. The first is pre-molecular and non-molecular. And then I would like to talk a little bit about the intraspecific variation. And then just chat a bit about the challenges we have encountered and a few words on future perspectives. So those black greens, it's not unfamiliar to you now. And at a very early stage, and people really don't know what are they. And they think some hypotheses said those are sunburn or sun-baked greens. So it's like a normal green got sunburn. Or another hypothesis is excessive formation of saps or even mutation of grains, all kinds of hypotheses. And Geoffroy, the person of the plate, this person, he recognized ergot as a separate organism from host plants. And his observation was supported by DeCandle. That was about 40 or 50 years later, and he gave the fungus a name, Sclerotium clavus. And it was another 40 or 50 years later, Tulassen discovered the sexual stage of the fungus. And then he erected the name of the genus called Claviceps. Meanwhile, he recognized 3 species. This here is his original jaw. It's just a beautiful, stunning jaw. And he recognized those species based on morphological differences, host, and also biological differences, such as microcephala, as he described, produces very long, very narrow stipes, and also tiny, soft head we call capitula. And on the other hand, C. nigricans, the distance between the paracesia, as I show you here in this red arrow, and are more distant than others. So that's his observation. And in terms of host, nigricans is on Sigus. There's Sigus, and C. microcephala is on Phragmites and Molinia, and those are the grasses in the wet habitat. And on top of it, and he also noted that biologically, microcephala, C. microcephala is different, and the sclerotia can germinate right on the plant as he draw here, and it doesn't need the dormant stage and or even the freeze shock like other sclerotia did. So after Tulassen, and in next 80 years about, and the Claviceps was described here and there. And so this is a list I summarized from Michael Bank and Index Fungorum. I might miss some, probably when they first described the fungus not in Claviceps and later on moved into Claviceps, but more or less it's this list, and it's a not long list, I will say. And also the species in red font were later on moved into other genera by authors, coming along. And in 1940s to between 1940s to 1950s, London in Australia did intensive work on Claviceps, and he got his PhD actually on working on ergot. And his dissertation is still very valuable reference for ergot taxonomy. He studied 25 species and did a very careful account for the morphological difference, host range, geographic distribution, and even the synonyms. And he described 8 new Australia species. So in his opinion, the shape and the color of sclerotia are informative, like here, the different color, but the size is not. And for the stromata, the color of a stipe or the head capitula are informative, or the placement of paracesia on this big head. And they can either be embedded or elevated. These are informative in his opinion, but the length of step is of little significance, as he described exactly the same way. And then he did mention some other special characters, such as a collar around the base of capitula, or a white ring of hyphae at the base of capitula, or loose hyphae on the capitula. I noticed those species, and I wish I could find a picture of them, but I couldn't find from in his dissertation. I guess we just have to pay attention to these characters when we do our monograph. And and the shape of size of Canidia are different between different species. However, within species, it's also quite variable. And so he concluded the Canidia size and shape is informative only at a certain level. And Lovelace in 1950 he did a very careful examination on for the Canidia from honeydew, and collected from 34 grassy species from Rhodesia. Rhodesia is a place, as I showed here in the map, the green spot, this green spot, and nowadays equivalent to Zimbabwe. So what he did is he collected the grass with ergot, and he randomly picked 25 ergot, and then he used a special tool to outline those sclerotia, and then he summarized them in terms of size and shape. Then he came up with these 13 groups. Now he can match six species already described in Rhodesia to the group, such as Paspali. C. paspali belong to group 2, and C. digitaria belong to group 5, and so on. So he also predicted that there should be 7 more species undescribed in Rhodesia. And actually, after this publication, he did publish a couple of papers describe new species from Rhodesia, but I don't see he matched his group, this group, to in the Canadian description, and for some reason maybe they don't match, or some reason I don't know. And another name you can encounter when you look at literature in Claviceps is Tanda. Between 1970 to 1990s, he described a couple of species in Japan, but he described tons of varieties. One of them, like this, I show you the picture here. This one is on Phalaris, and he did a very careful examination on the shape and the size of sclerotia and the stomata size, and even the paresthesia and the shape, size, very careful, and also the Canidia. And he did noted that this Canidia, you see there is a note here, this is Canidia from Phalaris, this is from Allopercuris. And he said this variety has produce a very big Canidia, but this is not convincing enough for him to describe it as a new species. So he called it variety. And based on just in the same way, he described tons of varieties. And whether those varieties belong to actually represent a new species, or truly belong to variety of some species, need to be evaluated. And a researcher right now in Japan, Tanaka, I don't have a picture here, and he is working on that. And also on the side note, this variety actually based on right now the molecular evidence, it's a new species. So, but meanwhile, it also tells us how difficult it is without the molecular marker to only depend on the morphology to tell apart the species of Claviceps. And I think probably that is also the part of the reason why there are not so many species described. And another character like sugar content has been used for described species from 1970s, and such as purpurea fusiformis, some species are rich in glucose, fructose, and fructose, and for other species, they are rich in sugar, alcohol, and also other species, they are mixed, have all kinds of sugars. And alkaloid content has been used as the character described species since 1950s. Even nowadays, we still use, and with even more detailed separation of the profile. And as I coded here, you know, the different species produce a different set of alkaloids, as just now Anna showed you in her photograph research result. And so in molecular error, when we talk about molecular taxonomy, it is not that we only use molecular evidence. It is actually the combination of molecular marker with other evidence such as morphology, phenotype, and ecology, or other physiological characters. And we call it a polyphasic approach. The first group use molecular markers is Pazotova group, and this group from Czech Republic had made a big contribution in Ergot taxonomy. And so in 1990s, they used RAPT and ITS1 sequences to characterize the different species. And also in addition, they developed a media. They developed a media called T2 media. So usually the colony of Claviceps, they grow on PDA, will all grow a lot of arrow hyphae and very fluffy, look fluffy, and they look almost the same. But on these T2 media, the colonies can show different characteristics. They are more compact, so different color and different textures. And you can see the pictures here. And show you different color and different. And also on this media, the colony produce a lot of conidia, and this provide opportunity for them to find more features, such as the presence of micro conidia, like this one on the left, and those little oval-shaped conidia, much smaller than the regular conidia. Those are the macroconidia. Some species produce them, some species not. And they also noted the difference or different patterns of conidia germination, and they call the microcycle. And some species, you know, the conidia germinate and then produce another conidia, and then produce another conidia. And then the pattern, you know, how many times they can produce this conidia, secondary conidia, and it's different between species. So they use that to describe the species. And the use multi-gene analysis for differentiate species is by a US group in 2008. And they studied the samples within the pre-molecular concept of Claviceps purpurea, and they recognized 3 groups here, not indicated G1, G2, G3. And G1, they said they are correlated with ecological niches, and G1 is mainly on terrestrial, and G2 is wet, shady places, G3 is salt marsh. And so until 2015, again, the Pazotova group translated that research result to formal taxonomy. And G1 stand is the C. purpurea sensu stricto. G2 is C. humidifera, and G3 is C. spartinia. And meanwhile, they also found another lineage and described as new species as C. arudinis. And they think the C. microcephala actually is a synonym of this. And in terms of the name, and they had a discussion about that. If you're curious, I'm not going to spend a lot of time why it's called arudinis and not microcephala. And you can always come back to this, to their manuscript to see why. And so this species separation elimination within and was supported by another study by Norwegian group. And they also used in the alkaloid profile to characterize those species. And however, their research results challenged the host specificity and also the ecological niche association. And more recently, a few more species were was described by this Czech group and from the species from South Africa. So in Canada, in our national mycological herbarium, we have over 700 specimens, and 90% of them are labeled as C. purpurea. And this is not surprising because actually there is no taxonomist actually seriously studied the Claviceps, but we do have some species described, such as by Canadian botanists, such as Grohii and also C. zizania. And we also have a few other species. And from 2016, our lab studied using multi-gene approach and a polyphasic approach to study the samples collected by all the resources other people collected for me and myself. I also collected some samples and from agriculture area and non-agriculture area. So in this multi-gene tree, we recovered all the 4 species described by the Czech group. And meanwhile, we also found a few new lineages. We described them as new species, such as this one, Siripicola, and because it's collected from riverbanks, lake banks, sand dunes, and it's quite common actually in North America. And both myself, Jim, and Steve, Kirk students collected one sample, and so they are mainly on Phalaris or Amorphila. And another species, this one I collected from Newfoundland, from the salt marsh. And this species had a very unique feature of a colony you can see on this right side, and very purple and very compact with this root system-like feature on Petri dish, and very different. So C. cubensis was collected by Jack Coyote, a research scientist in our center and from Quebec. And this species produce different shape of Canidia. You can see it's oval shaped, others are rod shaped or very different. And the last one is more interesting. This one, C. occidentalis, is from our cultural collection, CCFC. And the original specimen was collected by Dr. Campbell Bell in 1950s from Alberta. I think it's from Alberta. And so we went to the original location and tried to find the ergot again, but we haven't found it. And so we only have the culture. So it also tells us how important our cultural collection is. So in those liquid, in those nitrogen tanks, there might be more new species hidden there and waiting for being discovered. So I'm back to our chat, and this is updated based on our recent study. This is updated list of the species in Canada. And as I said, Microcephala is a synonym of Arudinis. And this total is 12 species. And more recently, the Czech group, they published more comprehensive study in the whole genus, and they separated the genus into 4 sections. And they also did a lot of discussion on the evolution of ergot, and in terms of host extension and all different aspects. And Kirk will talk more about this, and based on our new evidence, the genome studying evidence. And in the supplementary material of this publication, and they also listed evaluation of all the species described. They preliminarily accepted about 60 species, and should be evaluated more, but it really built a very good foundation for our monograph, and it's very valuable information. So Dr. Kalarick and myself, the guy stand here, he is Dr. Kalarick, and we are collaborate on the monograph of the genus based on this, built on this thing. And this picture was taken when I visited Czech just before the pandemic started, and it was quite a memory. So now I want to chat a little bit about the intraspecific variation. So remember this tree, even though we have a couple of species described all here and there and different species, and still almost 90% of samples still belong to this purpurea sensu stricto. So our question is whether this is just a pan-mixed population or whether there is some kind of subdivision. So this tree is generated by phylogenetic approach. So phylogenetic approach is based on the hypothesis that the relationship among the taxa is bifurcated like a tree shape like this. And it's not appropriate for a system if there are some horizontal gene exchange among the taxa. But for the network-based phylogenetic analysis, take that into consideration, such as BEAST, TCS, or split tree, and they are more appropriate for those systems. So we kind of try those methods, see what happens, and we use the split tree, analyzed the 156 samples in purpurea sensu stricto, and it come up 146 haplotypes. It separated into 3 genetic clusters. I call them GC1, 2, 3. And also these 3 clusters are also supported by other analysis such as BEAST. You can see on this tree very intuitively, GC1 is very deeply separated, and GC3 is embedded in GC2. However, you can see there is a gap here. Excuse me. So for PCS, and it's also very intuitive. You see the red dots, GC1 totally separated from other 2 groups. Other two group seems like there still is kind of gene exchange, but more or less they are separated. And a MOVA test, FST, and SNN, they all indicated significant genetic subdivision. And gene flow parameter, however, also indicated that there is a certain level of gene flow among the genetic clusters. So our question is whether those clusters, whether these different clusters are correlated with locations. So we mapped all the samples on this map, and basically they are from 3 different locations, and you can see, and on all these 3 locations and 3 GCs are coexist, which means it not correlated with geographic location. And we did the same thing with host group. We separated host as 2 big groups, crop TTC, because most of crop belong to TTC, and non-crop TTC and broomy, and all 3 crop clustered groups had 3 genetic clusters. So basically the take-home message is, so the purpurea sensu stricto is not a pan-mixed population. There is like some kind of genetic subdivision. However, they are not correlated with host, not correlated with geographic location, and there must be some intrinsic mechanism separate them. We hypothesize it might be mating type, but this need to be tested. And I think this should be a good graduate student project to work on it. And so now I would like to one minute and chat a little bit about the challenges we have met, and also a few words about future perspective. ITS ITS is a locus proposed by fungal community as a barcoding area. However, it's not very good for Claviceps. The first is the presence of these random big insertions. It's very difficult for the sequencing alignment, and there are also some area. It's very ambiguous. The alignment is very ambiguous, and it can be very misleading. And the second is low resolution. As I showed here in this red square box, and those groups can be separated by other housekeeping genes, however, cannot. They are all just in one group on ITS tree. And for third shortcoming is a non-orthologous copy of ITS. As I show you here, you see this GC4, and there is another GC4 here. And in the beginning, we thought this is a contamination, and we reassessed it. It's not a contamination, and it's truly a GC4. It's just a different copy of ITS. And the same thing happened for the G1. You see, there are two groups. G1 separated to two groups. So it's non-orthologous copy. This phenomenon was discovered in several of other fungal groups, such as Fusarium, and it can be misleading. And another challenge is localized disagreement between the protein coding genes, such as RPP2 and TEF. And for RPP2, you see this indicated G2 closely related with G21, but for TEF, is G2, it's G2B is in one. And also for other species, some species, RPP2 had better resolution, but for other species, the TEF had better resolution. And our follow-up research indicated both of the genes are likely under selective pressure, and possibly because they might be linked with some genes and positive selection. So they are kind of hitchhiking, or like a genetic undergone a genetic sweeping. And if you want to know more, and we discussed this in this paper. And so the good news is we have more genome available now. We can develop more markers. Genome, certainly genomics can answer all kinds of questions. And Kirk will talk more about that. But my question is, how do we use the genomic data for fungal taxonomy? And so first is what's the strategy to select the markers, select a panel of genes? And do we need a standardized panel of genes, just like what we choose ITS as a primary barcoding? Do we need a standard panel of genes for barcoding with genome data? And also, how do we handle genome complications such as ancient lineage sorting, hybridization, recombination, horizontal gene transfer? All of those, they can be all complicate the use of those genes for taxonomy purpose. And I guess we just have to really understand the genome very well and understand those complications, and before we can come up with a good panel of genes for taxonomy study. So with that, I thank you for your attention. So I will stop sharing my screen.
Thank you, Miao. Very interesting. We'll take some questions after the next speaker. At this time, we'll move on to Dr. Kirk Broders from the USDA ARS, and he's going to be talking on evolutionary insights into patterns of speciation and host range adaptation in the genus Claviceps and implications for disease management. Dr. Broder is a microbiologist and the curator of the USDA ARS culture collection at the National Center for Agricultural Utilization Research in Peoria, Illinois. During his before this job, he was a faculty member at Colorado State University, where his research focused on the ecology, evolution, and epidemiology of plant pathogens in both forested and agricultural ecosystems. And it was there that he began his research on ergot of barley and other grasses in the San Luis Valley of South Central Colorado. Always nice to see someone else who's getting interested in the ergots and has done some very good work. So at this point, I will turn it over to you, Kirk. Looking forward to your presentation.
Okay, great. Thanks for having me. I mean, it's been the previous talks have been great, and I'm going to try to combine some of that information because there is some overlap between the talks. And so the first thing I want to do is recognize the fact that the vast majority of this work was conducted by Dr. Stephen Wyka, who was a PhD student in my lab at Colorado State, and much of this formed the core of his dissertation. He has now moved on to a job at a company called LifeMine, where they're using bioinformatics to mine fungal genomes for biosynthetic gene clusters for therapeutic use. So I want to acknowledge him, and I'll acknowledge other people at the end, but a lot of this work was his, and I think he did a really fantastic job at trying to address you know, some of the issues we're dealing with ergot, at least in the San Luis Valley of Colorado. So just real quick, I wanted to sort of revisit the Claviceps purpurea sensu latu life cycle. You know, I think it's always important to keep in mind that this organism has such a broad host range, which represents lots of places for it to hide and lurk and infect and produce inoculum until it finally reaches a host of economic importance to humans like wheat, barley, rye. And as has been mentioned already, it doesn't cause mortality to the host, but it does poison the mammals that eat the grain. And so in many cases, this fungus does act like a defensive mutualist, where it is providing a defense from grazers to its host, and then the host is providing it with photosynthate and sugar so it can continue producing additional spores and reproduce. So in that light, that's how we approached this research. And, we try to think about, the evolutionary history of some of these interactions, certainly with Claviceps and its host. So as I mentioned, it has this huge host range. As Meow just mentioned, there has been some speciation that has occurred, but certainly in Claviceps purpurea, you have this expansive host range that is adapted to infect a number of different species around the world. And there was this really nice paper in PLOS One that came out a couple of years ago, which proposed this idea, well, not the first one to propose it, but certainly provided a really nice summary of data associated with this idea that Claviceps functions as a conditional defensive mutualist for its grass hosts. And there are other examples of Claviceps grass interactions that have had a positive effect for the host. A good example is Spartina species, that when there was a low infection rate, meaning not the entire head infected, you did see more seed and higher seed weight. And then there's good examples in fescue. Actually, this paper, the conditional defensive mutualist, was focused on grazing on Festuca rubra, and found that grazed fields had more ergot presence and ungrazed fields had less ergot, and that there may be some selection by grazers for grass that is not infected. Therefore, the only thing left is infected grain. And then there's been this really cool, interesting area. Anna touched on some of this earlier, and then there is a group in Germany, Hinch, that has looked at cytokinin production in Claviceps and how Claviceps produces cytokinin, which is a growth hormone. But based on the genome, it doesn't appear to be able to metabolize cytokinin. So it appears that the fungus has evolved this method to produce a growth hormone solely for its plant host. That growth hormone theoretically should trigger plant growth, and it should then also trigger increased nutrient flow to the seed production, and therefore the sclerotia that is forming in that floret. So there's lots of interesting interactions that's taking place, and we have to keep this in mind when we think about management, because it's not this typical gene for gene interaction like what we deal with rusts or tan spot. And it's even different than Fusarium head blight, which is it's sort of its own genetic plant microbe interaction that is occurring. And as also been mentioned, it's got, it's very closely related to Epicloid species, which we know are mutualistic grass species that can have a positive effect on their plant host. So from our perspective, so based on that, sort of the problem and sort of the impetus and start of all of this research for myself and Stephen was this increased ergot incidence in the San Luis Valley of Colorado and the Bighorn Basin of Wyoming. So previous to this, I'd primarily been working on wheat diseases. And the problem, so this is a map of barley production in North America. Sorry, I couldn't get Canada and the US perfectly merged. And there's a scale issue here, but in general, it's fairly accurate. So most of the North American barley is grown in North Dakota, Montana, Saskatchewan, and Alberta, but there's this little pocket down here in south central Colorado in the San Luis Valley where they also grow a fair amount of malting barley, and then also in the Bighorn Basin of Wyoming. And so they're sort of interesting little microcosms because they're fully separated from the rest of the barley producing regions in North America. And actually, after seeing the first talk, it was really interesting to see that ergot doesn't appear to be such an issue on barley in the Canadian provinces, but in Colorado, it is not a problem on bread wheat. There's no Durham wheat grown in Colorado, but it is becoming a bigger and bigger issue on malting barley here in the south central part of Colorado. So, based on, speaking with growers and agronomists to try to get a handle on understanding, the dynamics of these recent epidemics, we planned a series of studies that utilized some field survey work. We did some greenhouse inoculation experiments, and then, in a fantastic collaboration with Meow and Miroslav, we did some comparative genomics and pangenome research with some of these strains causing ergot. So what's so quite interesting about the San Luis Valley is that it is this little isolated spot in south central Colorado. So here you can see, we've got the Rockies, the Canadian Rockies that come down into North America, and then you have the Rockies here in Colorado. And the San Luis Valley is this little pocket right here. And it's quite isolated from much of the other growing regions in Colorado as well. And so what you can see here is you have the San Juan Mountains on the west and the Sangre de Cristo Mountains on the east. The headwaters of the Rio Grande River start here in the San Juans. And this is sort of a high desert, old lake basin. It's about 7,000 feet above sea level, but it sits on a fairly fertile aquifer. And actually the primary crop in this area is potatoes. It's one of the largest contiguous potato growing regions in the country. It's about 170 miles north to south and 70 miles east to west. I'm sorry, I'm not sure exactly what that is in kilometers right now. But also here on the east side, you have what's called the Sand Dunes National Park, which are the tallest sand dunes in North America. And it's really a beautiful spot. And so I've really enjoyed doing research down here. And you can somewhat see it in this photo, but these are largely center pivot fields. Almost all of this is under center pivot irrigation. And so the first thing we did is we just set out to do a survey because almost all of the barley grown in this area is malting barley, and it's all destined for Miller coolers in Golden, Colorado. And so we were working with an agronomist from Miller Coolers who sort of knew the history of the area and knew that there was pockets of the valley that historically would have ergot whenever the climate was good for disease development that year. And so we essentially started this surveys, and what we did is we divided up all of the different fields that we visited based on either a severe history, a mild history, or no history of ergot. And what we immediately noticed is that the field margins this is an example right here, oftentimes had really thick and heavy populations of brome grass, primarily smooth brome, which is an invasive species throughout much of the United States, and certainly in the Western US has become a big issue. And so a lot of our focus, came into the smooth brome, really trying to get an idea of how much inoculum is present in these ditch banks on these field margins. And so in this case, you've got a circular barley field, which is under center pivot irrigation. Most of these are 1 mile by 1 mile grids because the area is so flat. And then you have these ditch banks, which are right alongside the roads. And we were doing our transects through the ditch banks to quantify inoculum load in these areas. And what we found is that we had an increase in disease incidence over the course of the growing season. We surveyed in July, August, and September. What is interesting, so disease history, whether the field had a history of disease or not, was no, low, and high. And you can see that by September, this is in the ditch grass, not the actual barley field. By September, everywhere we could find at least small quantities of sclerotia developing in the grasses along the field margins, but certainly in areas where there was a high history, there are significantly greater quantities of sclerotia developing in those roadside grasses. Certainly, there was the severe ergot history sites have a large quantity of brome grass. So the importance value is a combination of the composition of brome and then also the quantity or the percentage of the area that was brome grass. And so just so you know, brome grass actually includes a number of brome species. It includes smooth brome, meadow brome, mountain brome, and then wheatgrass includes a number of wheatgrass species as well: western wheatgrass, intermediate wheatgrass, slender wheatgrass, quackgrass, and crested wheatgrass. I'm not a grass taxonomist. We had some help with this, but we just grouped them into these larger groups for simplicity's sake. And what you can see is that brome represents a very large and important source of inoculum in these field edges. And certainly, the fields that had a history of ergot correlated very directly with the amount of brome that was on the edge of the field margin. Wheatgrass was kind of intermediate in its proximity to no and high disease history fields, and the other grasses seem to be non-hosts. And then the non-host species were certainly more common around fields where there was no history of ergot. And then we actually counted the number of sclerotia in these 1 meter by 1 meter spots. So we built these square frames and we would drop them down and we would just count sclerotia. And what's really kind of scary is that in brome, when they actually flower, you can count sometimes over 80 sclerotia per head in many cases. And so you've got this huge potential for inoculum in the following season as well, not to mention honeydew early in the season. And so brome had significantly more sclerotia than the wheatgrass. And so if you take that average number of sclerotia per inflorescence, which was right around between 4 to 5, and it didn't seem to matter where the brome was. And then we multiplied that by the brome population, and you can estimate the sclerotia per square meter in some of these roadside ditch banks. And in some cases, in the fields that had a history of high level of disease, the estimated sclerotia per meter squared in the brome was sometimes 60 to 70 sclerotia per meter squared. So you've got a pretty significant inoculum source, and that's there year after year as the brome continues to reinfect itself. So what sort of comes from this is that brome acts as a very good reservoir for Ergot sclerotia. So you notice we don't have any data for barley here because we didn't see any major Ergot outbreaks in the years we were doing our surveys. There was some fields that we were able to visit, but they were not part of our survey. And so what we found is that disease in the valley is quite sporadic and oftentimes is related to scattered thunderstorms and rain showers that move through the area that could be driving the spread from the ditch banks into the field margins. So an interesting thing was, the fact that brome is producing all of these sclerotia. And so we also did some experimentation looking at the effect of infection on overall health and productivity. And we couldn't get brome to flower in the greenhouse, so we couldn't actually see if there was any positive effect on brome. But what we did find is that when we infected barley, there was actually a positive response to infection, low levels of infection, 5–15% of heads infected. We actually saw an increase here in both plant biomass and root biomass when barley was infected with ergot. And this may be a response to the production of cytokinin from ergot. However, we saw a decrease in seed germination. However, it was non-significant. And we saw no effects in wheat. So essentially, this is a neutral reaction in wheat. There's no detriment, but there was also no added bonus. That then moved us into trying to understand the evolutionary history of Claviceps. And so as Niall mentioned, there are now 4 sections in the genus Claviceps. And this came from the Pichova paper from 2018. And these are the 4 sections. And here is section Claviceps. And down here is divergence in evolutionary time. This is the entire genus diverging and increasing in species over time. This is section Bushel, and right here is section Claviceps. So you can see at this point, about 20 million years ago, something occurs that results in a rapid proliferation of species in section Claviceps and evolution to a number of hosts. And so we did a comparative genomics approach, sequencing the genomes of a number of members in Claviceps, section Claviceps, Bushel, Citrinum, and Passiflorum. And we looked at a number of things within the genome that may impact evolution. And one of those is transposable element content. And this has been shown in a number of species. And what is really interesting in Claviceps is it doesn't appear to have very many transposable elements. It's very low, especially in comparison to sections Bushel and Passiflorum. It's a really stark contrast, actually, when section Claviceps diverged. In the case of transposable element localization, certainly in section Claviceps and Bushel, the effectors are closer to transposable elements than other genes, so that would possibly have an advantage in potentially duplicating effectors and leading to effector diversity. There is also, we looked at whether or not different parts of the genome are evolving at different paces, which would indicate different selection pressure. And what we found is that doesn't appear to be happening in Claviceps. If you know the literature, this idea of a 2-speed genome is certainly been found in Zymoseptoria, which infects wheat, but doesn't appear to be the case in Claviceps. However, I will say that in section Claviceps here, in section Bushel, it does appear to be this trend and movement towards a potential 2-speed genome. But what does appear to be another really stark difference and could be quite important for the evolution of section Claviceps is the fact that Claviceps appears to have lost the ability to use RIP, which is this repeat-induced production. So what RIP does is it actually eliminates duplicated genes from the genome, which then allows the organism to continue evolving without these potentially detrimental duplicated genes. And so what RIP does is if a gene is duplicated through sexual recombination or duplicated through transposition, RIP actually eliminates that gene from the genome. And so what you can see here is in Claviceps, almost none of the genome is affected by RIP, but in section Pusili, Passiflorum, and Citrinum, RIP is functioning like it is supposed to and eliminating those duplicate genes. And so then what you see, the result is that Claviceps has a huge number of duplicated genes. And actually, Meow is doing a lot of this work right now on duplication of secondary metabolites. But you can see in section Pusili, Passiflorum, Citrinum that duplication does not occur. And when we look across all of the genomes that we sequenced, the blue bars here represent isolates in section Claviceps. And what you see is this is the percent identity. And so what you find is that, with genes that are 60 to 100% identical, these all represent duplication events. And so you have lots of gene duplication occurring in section Claviceps, but you do not have it occurring in these other sections. And these duplication events could be, duplication of effectors, duplication of alkaloid genes, duplications of entire biosynthetic gene clusters, things that could be contributing to evolution and divergence. And that's actually what we see. So in section Claviceps, which is right here, you have an increased number of and quantity of effectors, more secreted proteins, more secondary metabolites, more transmembrane domains. So this loss of RIP and resulting gene duplication may be allowing Claviceps to adapt to more hosts and more geographic regions by expanding secondary metabolites and effectors and transmembrane domains that are allowing it to infect additional hosts and adapt to climates. So our current theory, this is work that Stephen published in Genome Biology and Evolution, is that in the evolution of the genus Claviceps, you have this point where you start getting co-localization of transposable elements around predicted effectors before section Pusili and Claviceps diverge. And then at this point right here, about 20 million years ago, we think in section Claviceps, you have this loss of RIP-like mechanisms, you have lower transposable element contact, and then lots of gene duplication and an increase in host range. And so as a follow-up to that, we focused just on the Claviceps purpurea pangenome. And so for this, we had 24 isolates of Claviceps purpurea. These came from 12 different hosts, mostly from Canada and the US, a few from Europe and New Zealand. So it essentially we're creating what we call like a pangenome, which is, the genome of a species, but includes all of its variation at the same time. And somebody brought this up in Anna's talk when we saw this diversity in alkaloid production between different isolates. And that could very well due to the fact that only 62% of gene orthogroups that we found were found in all 24 isolates, whereas an additional close to 4,000 gene orthogroups were found in at least one or more strains, but not in all 24. And there was 1,000 orthologs that were only found in a single lineage. Now, many of these may not have an important function in infection or adaptation, but some of them very well might. And so what we did find is that here we have red is the core, this medium orange is accessory, and then this one is singletons. And you can see that predicted effectors, transmembrane domains, as well as right here, secondary metabolites are enriched in the accessory and singleton genes in this particular category. And so these are the ones getting duplicated, we think, and are being essentially diverging through sexual recombination and then flow between regions. There's pretty limited actually limited evolutionary selection on these genes so far, probably because many of them are novel and new, but there doesn't appear to be any selection force, which also supports this idea of more of a defensive mutualist theory. And then we looked at recombination rate across the genome as well. And what we found is that recombination rate is generally even throughout the genome. There are hot spots, but what we did find is that it's higher in genes associated with secreted proteins and transmembrane domains and lower in duplicated genes. And so these duplicated genes appear to be stabilizing in the genome. And as I mentioned, we did find some recombination hotspots. And I know this is a really busy figure, but I think the take home from this figure is that these recombination hotspots do not appear to be associated with any particular group of genes. You know, they're not affected with they're not associated with effectors, they're not associated with secondary metabolites, but they do occur in all of those areas. And a good example here is this IPS A2 and A1. So these are alkaloid genes that are right in the middle of a recombination hotspot. And so we believe that these were duplicated in a sexual recombination event. And, you know, this phenomenon could be then occurring in multiple other locations and also resulting in this increase in duplicated genes. So in summary, from the pangenome work, in section Claviceps, we see this adaptation to cool season, colder climate, and we think that recombination hotspots in both secondary metabolite genes, but also in other areas, may be leading to this increase in gene diversity in this section, which allows it to colonize more hosts and more geographic regions. And then there are, as Yao mentioned, there appear to be some cryptic species in Claviceps purpurea, and both the multi-gene phylogenies and whole genome comparisons, I think, are allowing us to start to dissect the evolution into more smaller climactic niches, whether it is mountain regions, coastal regions, salt marshes, and then we still have, the entire Claviceps purpurea sensu stricto that appears to have a very broad and geographic host range and seems to be maintaining, that pangenome of plasticity, which is going to allow it to continue to adapt to future control measures as well. We need to keep that in mind. So what have we learned about the ecology and evolution of Claviceps species? Well, we know that at least in the San Luis Valley, brome grass is a major source of Claviceps inoculum in the Mountain West, and probably in the US elsewhere. And controlling that inoculum is going to be very important. And I think doing surveys to understand that inoculum elsewhere would also be really valuable. Infection by Claviceps can have neutral to beneficial effect on wheat and barley. And I'm so disappointed we never got around to doing inoculations on brome, because brome is a rhizomaceous grass. So it grows by rhizomes and roots. And you can see that data from barley, that if you get an increase in root biomass and above ground biomass, that's going to allow brome to continue to colonize. And so there may be some evidence that, brome is continuing to colonize in these higher elevations where it's more poorly adapted, because it continues to get reinfected with Claviceps and can continue to grow as rhizomes and expand its area. The lack of RIP and subsequent gene duplication had an incredibly important effect on the evolution of section Claviceps and its ability to infect so many different host species. And then these recombination hotspots, I think, are going to may have resulted in this diversity of secondary metabolites, and also resulted in this variation of secondary metabolites and alkaloids in individual strains of Claviceps, as documented by Anna previously. So what does this all have? How does this all affect management, which is also asked about earlier? And the question really has to be about, how do you manage a defensive mutualist? It's not as straightforward as a true pathogen that operates in the gene for gene mechanism, where you can breed for host resistance a little bit better. because there really is limited host resistance. And I think any host resistance is going to be related to host physiology. It's going to be related to the structure of the head and structure of flowers and limiting the ability of spores to actually enter. So it's going to have to be sort of physiological protection. Fungicide application could be effective, but there's a big asterisk there because it needs to be timed precisely. And sometimes that may have to do with whether or not it's going to rain or you're going to have a significant rain-wind combination weather event. And even then, they haven't shown to always be effective. And when you have this huge reservoir of potential honeydew in field margins and ditch banks, that could represent multiple applications, which then becomes cost prohibitive because of the cost of the crop. And I think really cultural management and understanding what your inoculum reservoir is. Certainly what's your inoculum reservoir in neighboring grasses. Somebody brought up cover crops. That would be interesting if somebody would start doing surveys in that area. So you would know if that was a potential inoculum source. And then trying to quantify that and then, ensuring that you're getting rid of that inoculum or at least mowing down grasses if possible in high risk areas. So I think it's going to be, a multi-pronged approach that just simply requires a little bit more investigation and survey and monitoring work on the behalf of scientists and agronomists and extension specialists. And so, yeah, I think, over women, the other thing I didn't talk about is the sclerotia. So there also has been some debate about sclerotia in seed source, but I don't want to get into that debate because there's not a lot of evidence. Just a lot of anecdotal evidence, I suppose, but that is also something certainly to consider. And I think those neighboring grasses are really of a lot of importance, but have never been given much attention. So with that, I'm going to go ahead and finish up. I want to thank once again Stephen Wyka, but then also Stephen Mondo from JGI who helped us with some of the bioinformatics. Meow has been a great collaboration that she and myself and Miroslav have put together. And then Judy Jolly, who is the agronomist at Miller Coors, and Tyler Thompson is the farm manager at CSU. And this work was funded by the American Malting Barley Association. And then also Stephen was given a pre-doc award from NIFA to help with this work. So with that, I'll go ahead and stop my screen share and we'll open it up to questions.
Thank you, Kurt. Very interesting. I'll start you off with a question from Sean. Was there co-localization of accessory genes in subtelomeres or evidence of accessory chromosomes, or do you believe it is recombination driven and accessory genes are broadly distributed across the genome?
It's a really, really good question. I don't think our evidence is good enough to say yes or no to either of those. And to be honest, both could be happening. So I think our assemblies were good, but it was all Illumina-based sequencing. And I think some long read sequencing of a few strains would be incredibly valuable to fill in those gaps. Because I didn't say it because I think our evidence is well, it's shaky. It could be one of two things. Either there are actual dispensable chromosomes in Claviceps, which have been documented in other fungi, or it is a lack of our ability to assemble portions of a couple of particular genomes because we didn't have the reads or there's duplication events that allowed us to assemble those. So that's one possibility. The recombination hotspots, there aren't that many in the genome. There are some. So I think the duplication events are what is really driving it. And maybe Meow could comment on this. I know her work isn't published on this, but she's been doing a lot of work on duplicated alkaloid genes in Claviceps.
Sorry, I was kind of distracted by other things. So what's the question? I forgot. Can you remind me what's the question?
About the role of gene duplication in the expansion of secondary metabolite and alkaloid genes in Claviceps.
I see. Okay, so yes, yeah, Kirk and I, we were collaborated working on the Ergot alkaloid gene clusters, and we found a lot of gene duplication in in section Claviceps, and I think you are also asking whether they are located at the telomere or location, and I don't know at this moment. We had a long read sequencing, and yes, we actually I'm in the middle of assemble them, and some of them we can get almost chromosome level assembly, and but I haven't located the alkaloid duplicated, whether they are close to telomere. Yeah, I don't know if I answered your question, but I think in the literature, and they hypothesized, Shadow group and Carol Young group, they hypothesized that these genes are close to telomere, but we don't know yet. In the whole, throughout the genome, I mean in the genus Claviceps, we don't know yet. But it's a very interesting question.
Yeah, I'm going to ask you a question, Meow. Looking at this taxonomy, I mean, having taken plant pathology classes many years ago, I mean, we were just told if you see it in grains as Claviceps purpurea. Well, now under this, the new system, would we be correct that in the pathogen that affects cereal grains are mostly from the sect purpurea?
Yes, based on the evidence right now, I think if you find anything on the crops, 99%, I would say, based on the evidence we have now, probably it is C. purpurea. We haven't seen any of those other species, but keep that in mind, Jim. Remember, you also did the inoculation, right? And
Yep.
other species can infect. In the greenhouse, they can infect the crops. However, one species like that occidentalis, C. occidentalis, all the inoculation failed and they don't infect. Yeah. So in greenhouse they are possible, but in nature, less likely.
Yeah, no, and I think that's true. You always have to take lab studies with a grain of salt. There was also a question, and I'll leave this open to any of the speakers and to Dr. Turkington as well, as far as removing sclerotia from grain. How easy is that, and how could it be done? In fact, I might just turn that over to Kelly because I know he's had a lot of questions on that.
Oh, Jim, I was going to say I would defer to some of the sort of ag engineers that have worked in that area. I think there's a group in Saskatoon that have looked at FTKs and so on. But, you can use spiral cleaners. There's gravity tables and then the color sorters. So, I think they're all probably reasonably effective and would help to improve grade. I think probably the practical consideration would be what is the cost per bushel for the farmer, and it doesn't make economic sense. And if it means taking something from a number 4, number 3 because of ergot to maybe a number 2 or number 1, that would be a significant probably improvement in net income and would justify the cleaning. But good question, and I'll defer to the ag engineers on that.
Yeah, I would agree. I would also say from my experience that it's not 100% effective. But it can work, and you're right. If you can increase the grade, it could be definitely worthwhile from an economic standpoint. I have to admit, though, in my experience, what's disturbed me more than anything is often I find out that they're selling whatever they've taken out of that grain, like all the ergot, to feedlots, which I have tried to nicely tell them they shouldn't do that. But if people are wondering generally, how do you get rid of it? The best way is to bury it. If you've got all this sclerotia that you've taken out of dirty grain.
Okay, so
Pretty
that's
well.
the way we dispose the ergot, just to bury them. Do we need to detoxify, do something like a detoxify? No.
No, I realize that, and Cheryl and Sean may have more to say on this, but I think the problem is the cost to the grower.
I would say, like in a laboratory setting, we also want to minimize the release of grains and its spread. So, you know, we autoclave all of the biologicals in our program, and I think that much of the stuff downtown gets incinerated. Cheryl may have some comments as to what she does with her grain, but we autoclave all the biologicals afterward.
I see.
Jim,
Yeah, ours
sorry.
just go through regular disposal. With respect to the commercial aspects, I think Kelly gave some good information there. I think in terms of industry as well, we have to remember that the handlers, the exporters, they're also they're not just focused on ergot, they're focused on protein content, they're focused on other quality factors too. So they have a number of different variables that they're trying to manage at the same time as well as ergot. So that we need to keep that in mind, that it's a very complex situation.
I see. Yeah, as Cheryl, you are here, may as well I just ask another question. So do you think some kind of like PCR-based assays will be helpful?
Sorry, you cut out there. PCR-based assays would be helpful for what in particular?
So screening, for screening process.
I'm not sure. I guess it depends what you're trying to look for. Like, to be honest, if it's at the level of looking for presence of ergot sclerotia, I personally think that the visual inspection, I mean, if you're considering value for time, that's a very good way to do it. If you're looking at ergot alkaloids, then it has to be something chemistry based. I mean, for our monitoring and research, we're using like LC mass spectrometry based comprehensive instrument methods, but we're also doing research on looking at some new commercially available rapid tests that test for ergot alkaloids. We're seeing mixed results with some of those, but this is just preliminary stuff right now. So I think maybe Sean could jump in, and then if you're talking about screening for the fungi, Sean would be the one to answer the question about PCR, not me.
Yeah, sure. And I see that your hand's up there, Kelly, too. So maybe Kelly can jump in after I'm done. But we do high throughput screening of different fungi from the harvest through my lab. We have PCR based machines for doing that. Like Cheryl said, if it's presence of ergot, it's better to do that visually, or more cost effective, I should say. But if you are finding interesting things like duplicated genes that might be impacting toxin production, that might be something that would be interesting to include in future monitoring programs, to be able to assess if those genes are increasing in incidence or occurrence as the population structures of the pathogen change over time. And this sort of relates back to those genotype by management by environment interactions. So I do expect that the pathogen populations could be changing, and it'd be the best way to understand what's happening in those populations would be through PCR tests. And that's something that we have discussed. And I think it's the work that you guys are doing that's going to help lay the foundation to be able to do those types of tests, right? And we'll do some of that as well. Kelly, did you want to?
Yeah, it was to follow up on the earlier question about cleaning, and you know, the focus there obviously is in harvested grain, but you know, certainly over the years we've had questions, and I think Jim, you probably have also had these questions that you know, from the point of view of silage or maybe green feed and so on, and what impact is there. It was very interesting listening to Cheryl's presentation and seeing that your potential for alkaloid content in the honeydew seems to be quite low. So that may be a strategy to look at. Unfortunately, it would mean looking for honeydew in the field, and then maybe silaging a bit on the earlier side. But I guess the question that I would ask is, when does that transition occur? When do you start to see the alkaloid content increasing? Is that when the fungus starts to produce the sclerotial initials and differentiate into a sclerotium? How early does that occur? Because you could, silaging, you're usually around the soft dose stage, roughly, depending on producer and crop and so on. But yeah, that interesting stuff.
Thank you.
Oh, I'm sorry. Matthew Becker has asked, are sclerotia pure cultures, in other words, one strain, and does that mean that Claviceps purpurea is homeophilic? And I will leave that up to the experts.
Can I just jump in?
Sure.
A little bit. So as far as I know, as I read, and it's an unanswered question, and some people think it's homophilic, another group of people think it's heterophilic, and there were also like a discussion. And I think probably it's like it depends on which species and which, maybe even which strain. And overall, the answer is there is a discussion there, and no absolute answer at this moment.
I would agree. I think it's a question that I know I've talked about this with Sean as well, and it's something that it may be worth exploring that a bit to find out for sure what is actually going on there, but it would take a little bit of work.
I think probably this question can be started with the meeting type. If you want to get a graduate student study meeting type, and then this question will be part of the project, homophilic or heterophilic.
I will take advantage of all your expertise since you're on here. Being someone that's involved in trying to control this problem in Canadian crops, I know we have started working on some resistance, but after listening to some of your talks today, I'm wondering, what do you think is the prospect of finding durable resistance to Claviceps in these crops? And I should mention that the resistance we have found have been in wheat. We really haven't worked with other crops. And it seems to me that there may be differences among the different host species as to whether you can find this or not.
Maybe I'll
Anyone?
my
Yes.
2 cents in, Jim. So when we looked across the crops, we saw clear differences, and we know that rye is particularly susceptible. So I think when we're looking at durable resistance, I think there's probably is some level of resistance that's there, because we do see differences in susceptibility already, where we're seeing some crops are highly susceptible in brome grass or rye and these types of things. And we're already seeing from the presentation this morning that there is some genetics in the host that's impacting the response to the pathogen. And they talked about, the height genes as an example of that, which are around, gibberellic acid and some of these other components within cereals. So I think that it's probably going to be a complex resistance, and that'll end up being more durable. But yeah, it's going to be a multigenic trait would be my thought on that one. But I've been wrong before, so feel free to jump in if anyone else disagrees.
I think I will agree with Sean, and because as I also listened to the talks, and this pathogen actually is not pathogen, it's they are mutualism, and I guess it will be complicated. It's not pathogen. So yeah, that's my thought. So basically, the presence of the fungus is good for the plant. They are mutualism, right? And then how do you train the plant actually resist something good for them? Anyway, my 2 cents.
Oh, interesting question. You know, and it's good to hear those things. I mean, you're a taxonomist and look at it from a different perspective that I do as a plant pathologist. I will just mention, Kirk, we actually have a lot of brome grass up in Western Canada as well. And, you know, for those of you that are interested in doing something on a Saturday afternoon, you know, through June, July, and August, the way we actually go through sometimes to find out if there's infection in the brome grass is we just walk through the grass and let our hands trail beside our sides. If your hands come out sticky, now you can start looking for ergot because you've just found honeydew. So it is kind of an interesting way of doing it. Actually, my technician came up with that. Are there any other questions or comments that people would like to make or have? If not,
I see Gary Bergstrom just
sorry.
made a comment, and
Okay.
I think that's a good comment. It says hybrid rye seems to have much less ergot than conventional rye, apparently related to high pollen production and efficient pollination that outraces the pathogen. And I think that's a good point. I think, you know, I mentioned this in my talk, but I wasn't going to talk about it more because I'm not a plant breeder or plant physiologist. But when I talked to the wheat breeder Scott Haley, who recently retired from CSU, my understanding was, you know, one of the best ways is potentially to breed for particular physiological traits in flower formation or pollination that makes it a less optimal infection court for the spores to infect. So that also may be a potential route to decrease infection.
No, I think you're probably correct. I think there is some probable role of the floral structure and physiology. I will mention, though, as a plant pathologist, that it makes it a lot harder to do those assessments. So I think a lot of people have avoided it simply because of the amount of work that it's probably going to take. But is there something there? Yes, because we find the same thing up here. Hybrid rye tends to get less ergot than the male sterile lines. So there is definitely that role in there. I'll comment here, and Kelly may follow this up. Jill has asked, what about the role of soil plant nutrition plays in preventing ergot infection, in particular copper and possibly boron? I think there probably is an effect there. I think the problem you have when you have deficiency in some of these nutrients is you have more sterile florets. And the more sterile florets you have, the greater the likelihood of ergot infection. I don't think that, I mean, and crops should have proper nutrition. I'm not convinced that'll be a silver bullet, but that's just me. But there certainly are, there is room for looking into that. Kelly, do you want to comment on that as well?
Sure, Jim. We've certainly had that question and in relation to especially copper and boron. I would concur with you that it's not a miracle product, but if you have issues with deficiency, so you've done soil tests and you actually have deficiencies of those particular micronutrients, that can, as Jim has indicated, lead to issues with pollen viability. And if there's issues with pollen viability, a crop like wheat or barley that is more closed flowering and self-pollinating, those florets would open up because the pollen is sterile or has reduced viability in order to capture pollen that's floating around in the air. And that would simply provide an avenue of entry for those ascospores, or perhaps in terms of the honeydew stage and the conidia and insect activities. But if you have sufficient amounts of those micronutrients, adding more of them really doesn't decrease your risk. It's other factors. You got to keep in mind other factors too. So you get extreme weather events such as really high temperature or low temperature. That may also cause issues with pollen viability. Late herbicide applications and so on may also have the same effect, or other stress factors. So certainly with Ergot, it is an integrated approach, much like Fusarium head blight, that needs to be taken as far as management and ensuring good levels of copper and boron in the soil will help to at least mitigate that component of the risk for the crop.
Yeah, and I think along that line too, they've also found that your seeding rate can be important. If you can reduce late tillering, that will also reduce your ergot infection, because late tillers tend to have more sterile florets. So, I mean, there are relationships here between a number of things that can help reduce the effects or the infection of ergot to the crop.
And that's a good, sorry, Jim, that's a good point. We just finished up trial with Raja at Lethbridge with Ag Canada, and it compared conventional versus hybrid rye, winter rye, and seeding rate certainly was an important factor. So the level of ergot increased as your seeding rate decreased. So more tiller development and so on.
Well, it's 12:30. And I told the organizers we would finish at 12:30. I would like to thank the speakers for the presentations. I found it an extremely interesting workshop. I hope you all did. And with that, I would like to thank you all for attending the workshop, and I hope you all enjoyed it. Have a good rest of your day. Take care.
Bye-bye.
Thank you. Bye-bye.
Thank you.
Thanks.